Today we have David Lawrence from South Australia giving us a talk analysing and sharing genetic data with Python. Could we give David a warm welcome? Over to you, David.
Good morning. Hi, yeah, so I have a pretty weird career path. So I was interested in computer games at the start, and then I realized I needed more money and not working an insane amount of hours, and then worked in the corporate world doing Java programming for a while, and then went into bioinformatics, and that's actually been I looked at it today and I'm like, wow, I've actually been a bioinformatician longer than a programmer, so that was pretty strange. Yeah, but there's a saying in science where the best, as statisticians, they say the best thing about it is you get to play in other people's backyards. And I feel the same about programmers. So, if you're a programmer, you can, you can end up working on a national electricity grid or genetics. So there's a lot of cool things we can do. So the place I get to play is in biology, which I'm biased, but I think it's one of the most interesting things there is. So everyone, because of the vaccines, most people know about mRNA now. So DNA makes RNA, which makes protein. And they've actually, it was only in my grandparents' era, so maybe my parents actually in the 60s, they discovered how the DNA gets turned into proteins. So there's actually a code, it's called codons, and the bases get changed. The U is the RNA version of a T, but yeah, basically this is how it's translated. And basically the DNA gets translated 3 at a time into proteins, and then proteins, they fold up. That's a computational problem that people are working on. And it folds not just a single protein, it folds into multiple ones, then into complexes, and then it gets ridiculously complicated and hard. So there's a lot of computational stuff to do in biology. So yeah, and bioinformatics, when you think about it, the business requirement is basically to discover all about this stuff about life. And we know a lot, but there's an astonishing amount that we don't know. There's definitely lifetimes worth of work to find it all. Yeah, so there's a big demand for programmers, and the more Python programmers in bioinformatics, the better, I think. The better. It needs a lot of software skills. There's a lot of people are sort of self-taught biologists, and yeah, if you can come in with the software skills, you'd be very welcome. So yeah, check out the jobs. And if you're interested, my recommendation for getting into bioinformatics is this online web app. It's basically Leet Code for Bioinformatics, Project Rosalind. And yeah, basically it starts off really simple, counting nucleotides. So you basically just have to, they randomly generate an output, you run your Python program or whatever program against it, paste the answer in. If you got it right, you move on to the next one. Starts off easy, gets really hard. So lots of fun. So I'm going to talk about my particular job. So this is, I work in pathology. So here's an example of something that happens to me. So basically me and a massive team. So in the Women's and Children's Hospital, the other side of the river, generally, a baby might be born with heart problems. And the question is it due to infection? Is it due to a genetic predisposition? Is it due to birth complications, all kinds of troubles? And knowing the reason helps the clinicians decide what to do. So how pathology works is basically the clinician sends an order to the pathology company. SA Pathology is the public service provider for pathology in South Australia. We take blood, we take information, and we send back a report for that informs the clinician's decisions on what to do. So there's that's how it worked for things like blood concentrations of some calcium or something. It's a bit more complicated with DNA sequencing. So we still take blood, we extract the blood, put it in a DNA sequencer, process it on HPC machines, work out the difference between like a reference human genome and this patient's human genome, analyze it, and send back the report. So this is at Frome Road, the other side of North Terrace. This is my colleague and our expensive new sequencer. The way it works is basically the DNA gets put on this sort of glass tile, and they sort of wash the whole thing in bases, and when the next base at a time gets incorporated and it flashes a certain color, and there's a camera in the machine, and it just like basically sees that there's a million flashes of this color and that color, and it works out what it is, and eventually it turns it into DNA sequences. So this is the raw output of a sequencer. The new one we have is 16 to 20 billion reads in 44 hours. And these are usually like 250 bases long. The other things are like quality score and how confident the base calling is. So what do we do? How do we, 200 bases, what does that mean? The 200 bases, we work out where in the 3 billion human bases that came from. So there's like a string matching with a bit of fuzziness in there where we map to the human reference genome. So, and here's an example of a mapping. There's no letters in the map reads. That means it's a perfect match. But the G in the middle, that's a mismatch. So the patient differs from the reference genome. The reference genome has an A, the patient has a G half of the time. So you have 2 copies of chromosomes, one from mum, one from dad. And on one of them, we don't know which, either the mum's copy or the dad's copy, there's a G instead of an A. So this is called a heterozygous mutation. So where do these human genomes come from? It's basically was an extremely expensive project in the early 2000s. And yeah, you can basically download this off the internet. It cost insane amount of money to produce. You can download a text file. We use it in genomics for everything. It's extremely useful. Thanks for paying for science back, 23 years ago. We use it every day. And yeah, it was insanely expensive to do sequencing in the past, but it's actually much falling faster than Moore's law. So if you think computers are getting faster over time, well, genomics is going even crazier. And when things get cheaper, people want more of it. This is our demand for the stuff running through our software. That starts in around about 2013, and it's getting nuts, right? So good times. So how's Python used in bioinformatics? Well, generally, the tools for doing the hardcore computational stuff are written in C for speed, or sometimes Rust now. But Python's very commonly used to sort of join the multiple tools together. So you might define a workflow and coordinate the running of all these different jobs in Python. So here's an example. I'm just going to skip over this, but there's a program called SnakeMake, which is basically like make, but it allows Python. So it's pretty cool. Used in bioinformatics, maybe it's useful for you guys as well for dependency stuff. So for research, typically researchers work in small teams, and the output is basically a paper. So someone has a biological question, and you work with the biologist for 6 months to 3 years. And a lot of the time, it's stuff like, we have this data, we have a biological question, what can we do? And you often try a whole bunch of stuff, you write one-off programs, you investigate it, and Python is amazing for this because, I can write Python 10 times faster than C or something. I don't know what it is, but it's like, it's so much easier. And the thing is, we have HPC, we got hundreds of cores, we can run things in parallel. It doesn't matter how fast it is. Often we only run it once. We're just experimenting. And if we can experiment twice in a day rather than once every few days, we converge on the answer faster, and that's great. So yeah, to go back to variant calls, so the original sequence is at the top and the bottom one is at the bottom, that's called a point mutation. We have a file format for this, it's called the VCF format, and basically there's a chromosome location, a chromosome name, a position, and then the original sequence, and then the new sequence, and a whole bunch of other stuff. So I've written some software that's a use of SAPath to analyze this kind of data, and it's called VariantGrid. So what we do is we start with the sequencing, which has a huge amount of data. Then we get the diffs, and we do all this on an HPC. And then from this, the VCF, the variant calls, then it gets ingested into our program, and I take over from that point there. So it's basically a Django app. It's running on GUnicorn with Postgres being the central database. And whenever we have anything, more than a couple of seconds, I farm it off to a queue, and celery workers do the work. Most of the time, the longest running jobs might be, say, half an hour, which is for annotations. They run things like computational prediction. So, for instance, you might have a sequence, and it'll do things like look at the charges of the new modified protein and work out how much of a sort of how wonky the protein will go and classify it as pathogenic or something. And so there's all these different tools and public databases that we use to work out what a variant does. So yeah, here's the VCF again. So the way I represent it is in Django. Because this isn't in the Django stream, not sure if everyone knows Django, but basically you can declare classes that represent database tables. Here's my contig is just another biology name for a chromosome. Position, reference, and there might be multiple variants for a location, like, people might have a T, you might have a C, someone else might have a G. So you can have multiple ones there. And you can write your Python classes, and it sort of handles the SQL for you. So instead, on the bottom right here is the database, and on the top left is how you write the Django code. And it means that instead of writing all the technical computery stuff, you're just sort of working with variants and samples, and you end up sort of working in your domain, which is really useful, instead of computer code. Still computer code, but so there's a huge amount of biological data, and we get basically everything that we can find. We'll run it, and that there's hundreds. We have hundreds of columns of different tools and public databases that tell us what a variant does. There ends up being hundreds of thousands to millions of variants, and almost all of they're things like be a millimeter taller or shorter, or have a slightly different color hair or something. So you don't care about that when you're searching for the genetic cause of disease. So most of the work is throwing out all of that stuff. And to search through this is not easy. You have to know, it's like a mini research project. You have to know about the disease, how common it is, the family history. You end up going and looking through the literature. And so these a team of people called medical scientists who basically do this investigation. And the goal is basically of this program is to allow those people to run their own filters on huge amounts of data sets, because they've got excellent medical knowledge, but not so, they're not going to be able to run Jupyter notebooks or something. So a naive way to do filtering would be something like this, where you basically build up filters, but the trouble is we're often running something like 50 to 100 filters, and this gets unwieldy very fast. So basically, I have a mate who works in video compositing, and he showed me one day a directed acyclic graph. Often these are sort of tilted sideways, and basically the output of one node goes into the other one, some kind of filtering or thing happens, and that's how it works. And that allows people with domain knowledge to apply computational work, which is exactly what we want to do, so ripped it off and do it for genomics. So the way it works under the hood is each node returns a Django queue object, which is basically Django's way of filtering a query set or an SQL query. So you can take these queue objects, do logical operations on them, and make a SQL query. So here's an example. So I have here, there's I've got a whole bunch of queue objects. One of them is population frequency greater than or equal to 0.01. One of them is gene symbol in this gene list, and the other one is damage greater than 0.5. And then you can run, you could write this by saying, Q and Q2 and Q3, but you can also just reduce it down, applying and like that. And you can, if you want to have basically, apply this filter or this filter or this filter and this filter, you can build all kind of logical operations in this way. And this is what the GUI looks like. Basically, on the left is the nodes, the filters, and you click on one. So we've clicked on the red one in the middle, and that loads the population frequency, and you can choose what databases you want to use in your population frequency. And below that is the grid of the variants, like the 31 that appear in that node. And it allows the medical scientists to sort of jump around and like apply more or less stringent filters if something comes through or not. They can adjust the population frequency if it's not quite known how common a disease is, things like that. So it allows them to do their own filtering. And yeah, we try and allow the GUI to work nicely for the particular filters they want to run. And if you click on a variant, behind this loads a page per variant, and we have all that annotation details that you can view. Yeah, because of the data size, we have something like 30,000 samples, each with 1 million rows. We store everything in per patient Postgres partitions. And we also filter those. We might have multiple partitions per sample. So 95% of a patient variants are common, but a lot of the time, if it's a rare disease, it happens 1 in 10,000 people, you just want to throw away those really common variants. So by putting them in separate partitions, we can choose to jump to just the partition we're interested in, which makes it really fast. So here's an example where at the start we have basically collections is the name of the partitions that we're after. So we always apply the rare one, but then if someone asks for common variants, which means they haven't applied the population filter, then we also include the common collection, which is like 20 times as big. And then we do a filtered relation, which is Django's way of joining to a table with a constraint. And because of that constraint, like we say, just this parent table, just this one, and then the Postgres knows, okay, well, because of that constraint, I only have to look in this partition, and that makes the query really fast. Because yeah, if we didn't jump to the partitions, we're talking about billions and billions of rows, and that's not going to perform in real time. So once we found these variants, we have classification, and this is the classification part of the project. So basically, we auto-populate it, but the medical scientists can also tweak it, go grab information from various other sources. Some things you can't automate, like interpreting literature, well, not yet. And yeah, so ultimately what they do is they follow a framework called the ACMG guidelines, and they finally classify a variant in one of 5 ways. It's benign, likely benign, uncertain, and finally likely and then pathogenic. That means definitely disease-causing. And then we send that back to the clinicians, and they can look up drugs and stuff to use. So one of the troubles with this is that the health in Australia is basically run by states. And so everyone sort of works by themselves and don't talk to each other. So if someone in Victoria came in and there was a variant that was pathogenic, and someone in South Australia and they had the same one but they said it was unknown, then we would never know, and one of them got a bad diagnosis. So ideally we'd like to share, but it's actually really difficult because it's patient data, and there's all kinds of ethical and security issues to deal with that. So there's a group called Australian Genomics, and they sort of sit as a national group that sort of works above the states to try and help the individual states work better, especially better together. So they came up with the plan of Shariant, which is basically they have a central system where you take the state's individual classifications and send them up to a central server with all of the anonymity and all kinds of stuff that you have to deal with. And if there's a discrepancy, so 2 different states have different results, then it basically sends an email and says, look, sort this out. And then there's a little workflow that we send them down. So yeah, this project's been running since 2019, and so far we've linked up basically every public pathology provider in Australia and also just recently New Zealand. We're now starting to go after the private labs, pathology labs, and we're also starting to go into collecting the cancer ones, which are a little bit different and require a bit. So the ones we've traditionally captured are the inherited ones, and we're starting to capture the cancer ones, which are, have a bit of different information. So the way it works is basically, so SA Pathology, they use VariantGrid, and so we can do whatever we like there. But in other states, they use different software, and so we sort of have to write integration software with them and talk to their system, and then translate the data from their format into the common format, and then send it up via API. And then if two different labs get their different results, then this is what we show them, a diff. And then they they usually have a meeting, bring in the clinicians and the big guns of their labs to decide what happens. And then they sort it out, go through a workflow. Ideally one of them changes to agree with the other one, and then it eventually cascades through their system, through the API again, and then we mark it as resolved. So yeah, one of the troubles is how to handle data from many labs. Every state does their own thing. And so we had to write custom converters to put it into JSON. So sometimes we run it on their systems. Sometimes they just dump data on an S3 bucket, and then we run the code on our cloud system and send it from our cloud systems back to another through the API. And yeah, so we allow basically everything. We allow people to give every bit of information they have, but after we've investigated and asked for them what it is, then we can sort of add a bit of type information, such as float or int, and we try and get them to use a controlled vocabulary, like an ontology. So if a lab calls it population frequency, another lab calls it pop freak or something, we will convert that and send it all up on that, with the consistency on the API. So we try and hide the differences in those connector programs. So here's what the JSON looks like. And the advantage of this is we have, I don't know, maybe 100, 150 different fields that we collect from all the different labs. What we do is so here there's nomad allele number, which is how many counts this has been seen in a 100,000 person population survey. We know that's going to be an integer. So we have basically a key with an integer on it, and when we get the data, we can run a validation and say, this is supposed to be an int. It's a string. And we can raise a little warning and stuff like that, which just helps keep the data pretty clean. So yeah, here's the project timeline. So basically, I started this. So I work as a researcher on genomics, and people kept coming to me asking for me to do filtering, and I said, why don't I write some software so that people don't bother me anymore? And then it got used, and then the researchers started using it. Then the diagnostic people started using it. We ended up putting, basically we won a project to sort of, people have this very rare disease, and they basically have this online site where all researchers from around the world can upload their data and share it. And we're getting external funding from the States, and that's still ongoing. That was 2018, the first funding, and then we hired. Then we won the so Shariant was a project that we had that Australian Genomics wanted to do, and VariantGrid won the tender for that, and we used the tender for that and used VariantGrid technology as the base for Shariant. So we sort of have the ability to skin it and turn on and off things through settings to make the site work however the client wants it. And swap the CSS out and stuff. So in 2019 we got a second developer, which was amazing. And 2020 we went open source, and you can go to the GitHub page and check it out. In science, you're supposed to write a paper to advertise your project, and I've been I don't know, I'm a programmer, not a writer, and my boss keeps on my back to write a paper, and one day I'll get to it. but yeah, and then so it got diagnostic use, and it's been used by SAPath, and yeah, so that's how it's going. So thanks a lot. Cheers.
All right, thank you very much, David. What a great sequence of slides. Can we have a round of applause for David? Now, we do have a few minutes for questions, so if anybody in the room wants to put their hand up, or if anybody wants to type their questions into Discord and we can read them out here, please do so. I'll give you a moment to type things into Discord, because I know it can be hard to raise your hands sometimes.
And I'll check and answer them later if anyone else has anything else. So. But yeah, feel free to ask any questions.
All right, we have a question from down here near the front.
Yeah, I guess I was just wondering how kind of easy and usable you find the kind of ecosystem of data APIs like that NCBI provides for looking up your sequences and stuff. Because I've made my way slowly into this world, and yeah, I found it a struggle.
Yeah, okay, that's a great question. There are tools. There's like on Trez, there's a Python library, BioPython, and that sort of manages that. We use that for things like querying, say, how many publications there are for a gene, or when was the last time, or other things. But basically, the tools aren't awesome. We write, basically we always wrap our API calls in with timeouts and exceptions because the services are often down. I don't know. It's but yeah, it's a bit of a weird. Basically a lot of work was done a long time ago by people that there's a lot of somewhat questionable decisions that you wouldn't necessarily do it this way. Now, and but yeah, it's a big, that's why we need more programmers in the field. Sort our APIs out, and there's a lot of custom file formats and wacky old, really inefficient old file formats that are there instead of, in my ideal world, every service would have a REST API, and it would be trivial to get everything, but that's not a world we live in yet. So. But yeah, more programmers would make that world a reality, so consider bioinformatics. Yep.
All right, we have one question from the Discord. Do the findings push to or pull from open targets genetics?
Open targets genetics? I'm not sure. So we go to ClinVar as well, which is a very common, that's the most popular as far as I know, variant classification sharing platform. And we collect a lot more data than they do, and we also collect stuff that hasn't sort of been approved for public use. So it's behind like a private server and allows the labs across the country to sort out any problems out of internally, and then after a while they've sort everything out and then they share it, and then once we sort of set things to the public share level, then we go through and collect everything and send it to the external databases for everyone else to be able to look at. So we do talk to other databases and push it out, and we also pull theirs in so we can do comparisons if ours are different from theirs. So we both push and pull to external databases.
All right,
Yep.
That's all the time we have for questions. I have the traditional speaker's gift for David, the wonderful PyCon AU speaker's mug.
Thank you.
One more round of applause for David.